Again, welcome everybody. This is our talk on how plants have microbiomes too with Carrie Harwood of the UW. I'm Elizabeth Gage. I'm the WNPS Office and Volunteer Coordinator, and I'm going to be fading into the background pretty soon. But I would like to let you know we will be taking questions at the end of the presentation, and you can put your questions in the chat area. I also check the Q&A, but it's helpful and great for me if they're concentrated in the chat area. So go ahead and put your questions or comments there. We will be taking the questions at the end. And I think that's about all I need to say. So I'm going to introduce Yanka Hobbs, who's the current CPS Central Puget Sound Chapter Board Chair, who's going to give us some news about the chapter. And while she does that, I'm going to share my screen.
Hi, everybody. The first order of business is pertinent to everybody. The WNPS 2021 calendar is available now through the website. And since we're not meeting in person, it's probably the easiest way to get one is to order it online. And also, since chapters are unable to sell calendars at events, they've set it up so that your chapter gets $3 from each calendar sale if you're a member. So and of course they are beautiful calendars and informative, and you need one. And they also make good gifts. So after the presentation, go buy yourself a calendar and one for all your friends. Chapter news. We are planning to have our annual holiday get together. It's going to look on December 3rd, 7 p.m. It's going to look a little bit different this year since it's going to be on Zoom. And we will have maybe a couple of quizzes with prizes, hopefully some time to chat and socialize, and our annual chapter awards, which we always award a member of the year, a steward of the year, and a professional who has been particularly helpful. And if any of you have an idea of somebody who might deserve one of these awards, please send their name in to the office with a short description of why you think they are so deserving. And now I will pass the baton to Shelley, who is the program chair and organizer, and responsible for finding also all these interesting people for us to listen to.
Let's give Sharon Baker some credit too, as we share that position. Anyway, tonight I'm very pleased to introduce Carrie Harwood. In her own words, Carrie is an expert in the care and feeding of bacteria, especially the kinds of bacteria that like to live near, in, and on plants. On the other hand, she claims her knowledge of plant biology is weak. She says she took a botany course 45 years ago while in college, but hasn't taken any coursework in botany since. That's why she likes her project so much, studying the interactions between Populus or Cottonwood and its microbial community. It is an opportunity to keep learning. Carrie has been a professor in the Department of Microbiology at the University of Washington since 2005. Before that, she was a professor at the University of Iowa for 15 years. She says a large part of her time is devoted to running her research lab overlooking the Montlake Cut. Although it's hard to tell over Zoom, I know all of you will join me in giving Carrie a warm welcome. Thanks, Carrie.
Thank you. Let's see. Well, here's what I look like, but we're going to quickly segue over to share screen. I'm going to turn off my video. And let's see. Is that sharing? Can you share?
We
Can
see
you
your
hear?
name.
Can you see?
Bottom green button.
Yeah. Wait. Oh, I see. Yeah.
There it is.
Yeah. You can see it now?
Yes.
Okay, great. So, of course, I'm not on the first slide. Let's see. Okay, hold on. There we go. Okay, so plants have microbiomes too, plant microbe communities in cottonwood trees, so which are also known as populus, as I'm sure you all know. And cottonwood trees, as I'm also sure you all know, are all over the place. I didn't realize they were until I got involved in this project, and then I'm like tripping over cottonwood trees. So I want to talk about a project that we're doing at UW in as kind of a subcontract from Oak Ridge National Laboratory, which is a Department of Energy national laboratory. And that's why all my slides have the Oak Ridge logo on them. So there are bacteria, they're really tiny. Bacteria are really tiny. And I'll get back to that in a few minutes, but they're so tiny that we can't see them, but they're all over our skin and our bodies and in our bodies and on every surface that we touch. And of course, humans have their own microbiomes. We have our own set of bacteria that like to live on us, all over our skin. We've got tons of them in our intestines because they help us digest our food. And our lungs are pretty sterile, but every place else pretty much has bacteria all over them on our bodies. And then plants are the same way. Plant roots really have a lot of bacteria living around them because the plants release nutrients and the bacteria are attracted to the nutrients, and the bacteria even go right into the plant tissue and live in the plant tissues. And we know that microbes, of course, the plant helps the microbes by feeding them, but the microbes do things for the plant. They help the plant acquire nutrients. For example, they solubilize phosphate that's in the soil so that the plants can take it up. Plants need a lot of nitrogen. All living things need a lot of phosphate and a lot of nitrogen. And we have a lot of nitrogen in our atmosphere, 78% nitrogen gas, but we can't use that form of nitrogen. And so bacteria convert the nitrogen to a form that plants can use it. A few bacteria can do this. We can also fertilize plants with nitrogen that's made commercially, but that doesn't really help in the wild, and we shouldn't be doing that anyway. And sometimes some bacteria produce hormones that affect plant growth. And then some bacteria protect plants from pathogens that would infect plants and cause disease. So there's all kinds of stuff that bacteria do, but we've really just know the tip of the iceberg. And we know that plant and bacteria talk to each other, but we don't know very much about their communication, their lines of communication yet. So what you'd want to do is to understand the plant microbiome well enough to be able to control it so that it would help the plants that we're interested in to be as healthy as possible. And this is a photo of Populus, a little piece of Populus root. And these green things are bacteria. And you see bacteria all over the Populus root and even inside the root. It's kind of hard to tell here what's inside and what's outside, but there's a lot of bacteria colonizing Populus roots, we know. So things that on this project that we work on, which is called the Plant-Microbe Interactions Project, it's really fundamental research. We want to know how the plant host, a populus, recruits the microbiome and then responds to it. We want to know the microbiome, which is the assembly of microbes that live with the populus. There's like lots of different kinds of bacteria. So how do they know, how does the populus assemble its own microbiome? That's really different from human microbiomes. How does it assemble that specific microbiome? And how do the two talk to each other? And how does the microbe change their physiology in response to the plant? So we need a multidisciplinary approach to do all this stuff, you know. So people that know a lot about plants. The reason we're involved is because there weren't too many people at Oak Ridge National Laboratory who knew about microbes and bacteria. So they asked us to join in. And we're kind of experts in microbial communication. And then there's also a group at Duke University that studies fungi, and they're involved too. So Sharon Doty, who's at UW and knows like a ton of stuff about Populus, and gave me this photo she took. And so these are cottonwood trees, Populus growing alongside, I can't remember which river this is, but this is in Washington State. And so they're in the willow family, the aspen family. And one thing about them is they can grow in really marginal soils. And so you can grow up lots of populus if you want to use them like as a biofuel plant, for example, and not displace the land that you'd be using to grow crops. And also populus grows very fast. So I mentioned that populus is a good biofuel plant. And this is something that the Department of Energy is interested in. So they want plants, so you can grow up a plant, harvest it, and squish it all up, and get its cellulose, which is basically what they're made of, exposed. Plants do a lot to protect their cellulose, but if you can break down the protective things in the cell wall and expose the cellulose in a plant to enzymes and things that break down the cellulose, then sugars are released. And then special, again, microbes, which might be engineered to do certain functions, can convert the cellulose and other components of plant tissue into biofuels and other products that are valuable. And so the idea is to make biofuels that you can add as an additive to gasoline, like ethanol, so that we would have a sparing effect on our petroleum industry, not have to use as much fossil fuels. And also, of course, plants use CO2 when they grow. And so that's mitigating the CO2 buildup in our atmosphere. And when we burn these fuels, the CO2 gets released again, but it's supposed to be sort of a carbon neutral process. So Populus is one of the biofuel plants that the DOE has designated to have in their sites. And they, the Department of Energy has paid to have the Populus genome sequence. So we know the whole tree genome. And it's also established plantations, or they call them common gardens of Populus, at 3 different sites in the Pacific Northwest. There's 2 sites in the state of Washington. One's near Vancouver. There's a site in Oregon, and I think there actually might be a site in British Columbia, where they grow all different varieties of Populus, because they want to develop ones that have kind of weakened cell walls so they can be broken down easily. Of course, you have weakened cell walls, so you're going to get infected by pests more. So there's a trade-off there. So I just want to talk a little bit more about the Department of Energy, which is quite a big, you don't hear about it too much, but it's quite a big agency, $36 billion a year. And almost half of its budget is devoted to sort of maintaining our nuclear arsenal. And so a lot of stuff that's done at the DOE is top security. The DOE used to be called the Atomic Energy Commission. So they branched out a little bit from nuclear weapons, and they do energy research and development. This is quite applied research. It's a lot of research on batteries, on materials that would be used in solar panels, stuff like that. All kinds of energy related technologies. And then, of course, as a result of the nuclear arsenal, they have environmental cleanup responsibilities. For example, at the Hanford nuclear waste site, and a lot of money is spent every year on environmental cleanup. The Tri-Cities wouldn't exist if it weren't for the DOE. The amount of money, which is billions, that the DOE spends to clean up the Hanford site. And then there's basic science, which is where I sit, which is just sort of things you learn and you don't really know if there's going to be a specific application, but you kind of couch your research in the idea that there will be an application at some time. So it's sort of the seed corn of our technology development in the US. You know, some of it will be useful, some of it won't, but it's all interesting to people like me. So for most of you probably already know this, but in case you don't, that we do a lot of research in the United States, and it's mostly paid for by the federal government. And most of it is done at our research universities. There's about 100 research universities in the US. And of course, the University of Washington and WSU are 2 of them. So the idea is that the professors do the research and they write and get grants from the government, like from the National Institute of Health or from the National Science Foundation. There's a certain amount of Defense Department research that's done. And the UW takes in $1 billion, over $1 billion a year in research funding from the government. So it's, you know, it's really exciting for that reason to be a professor. It's very dog eat dog and competitive to get the funding. But when you get it, it's like, it's great. So like the best job in the world. So these are, but the DOE is different because the DOE has national labs and they tend to put their money into the national labs and then they fund some university research, but directly, but a lot of it's funded in collaboration for people who work in collaboration with people at the national labs. And characteristics of the national labs are, this is where like the big science gets done. These are where all the nuclear accelerators are national laboratory facilities, like Brookhaven National Lab, Argonne National Lab, the Fermi National Accelerator in Illinois, and there's one at Stanford, but it's owned by the DOE. And there's other big instrumentation like supercomputing that's done at the national labs. And so you notice that the national labs are not so much in urban areas, and there's political reasons for the placement of each of these national labs. But we're fortunate to have one in the state of Washington, which is PNNL, the Pacific Northwest National Lab. And I do some work with them, but now I'm talking about work that we do with the Oak Ridge National Lab. Some of these, again, are very high security, especially like Los Alamos and the Sandia National Labs, which are nuclear weapons security type labs. And these labs are very difficult to get access to. But usually if you're an American citizen, you can get access to the other labs. But if you're not an American citizen, even if you're a green card holder, you have to go through all kinds of paperwork to go onto the sites of the labs. So let's see. Oh, so this is Oak Ridge National Lab. And so they're big. The Oak Ridge National Lab and PNNL too have about 5,000 employees each. Oak Ridge National Lab was built here, I'm sure, partially for political reasons and partially because this is where the first nuclear fusion reactions were done as sort of a prelude to our atomic weapons program in World War II. And they chose this site because it's really hilly and it has these hollows, like Dolly Parton hollows, between the hills, and they did their experiments in these hollows because they figured if they had a nuclear explosion, it would be confined to this little area. So there's some history here, and you can see the original nuclear reactors, and it looks like incredibly primitive. Anyway, also the other thing that Oak Ridge National Lab has is the Summit supercomputer, which can perform 200 quadrillion calculations per second. And up until this summer, it was the fastest supercomputer in the world. But this summer, a Japanese supercomputer came online or was improved, so it's a little bit faster than this one. But usually, it goes back and forth between China and US on who has the fastest supercomputer. They do other things too. I don't know if fast is the most important thing. So now, this is, now I'm going to talk just a little bit about the actual project we do, which is the Plant-Microbe Interfaces Scientific Focus Area. And this is sort of a summary of the project. And it starts with populist trees. And they're like really big. and they have roots, okay? And the roots have fungi associated with them and bacteria associated with them. And so our plan, our overall plan was to break everything down into its component parts and then put it all back together again. So it's like if you have a car and you want to understand how it works, you take it all apart, then you put it back together again, and then you understand what part does what. And so that we do that kind of thing in science a lot. And so our plan was to go out and get microbes out of the roots, bring them into captivity, and then study them, study what chemicals they might make that they use to communicate with each other or to communicate with the populace. And then, of course, we wanted the genome sequences of all the as many bacteria as we could get. It's kind of expensive. And we had the genome sequence of Populus, because if you have the genes, then you can sort of read the genome sequence and understand a lot of things about the function of the organism that has that particular genome. So, so this is where we started. One of the guys at Oak Ridge was flying into the Knoxville airport, and he looked down and he was flying over a stand of cottonwood trees. So then a bunch of guys from the lab and gals, students, went out to near the airport to these populus, and they dug up the roots. They started near the tree and they went out and out and out so they could get these really fine roots. And they got the roots and then they sterilized them with bleach, because we are most interested in the bacteria that were living inside the tree. And then if we squished up kind of the inside of the root, macerated it, and then put a little tiny bit on an agar petri dish, then we'd get these blobs growing up. And that's where one bacterium fell onto the petri dish, and it grew up into, these are colonies or clones of the original blob. And the colonies, this colony probably has like, I don't know, like 500,000 cells in it. So when you get a lot of the bacteria together, you can see them. So we got, or Dale, who's at Oak Ridge and is actually used to be a student of mine a really long time ago, got 3,000 different kinds of bacteria on petri dishes like that. And then we sent them to this other facility, DOE lab, and in California, and called the Joint Genome Institute, and they sequenced a lot of the bacteria for us. So we had their whole genome sequences. And then what we did, or what Dale did and other guys and the other people in the project, was they took populus cuttings that were as sterile as they could get them, so they don't think they had too many bacteria in them. And they put them in soil, well, clay, sterile soil, and with different single types of bacteria. And then they saw how well they could grow in the tissue of the populus. So they'd put them in the soil, and then the populus would sprout into a little plantlet, and then they'd look in the leaves and see if the bacteria had gone from the soil into the leaves, which means they were all through the populus. And then we looked for the ones that were the best colonizers. And we kind of have focused on those to study in more depth. And so then we have to like, do stuff like get molecular tools so that we can go in and change their DNA around and et cetera, make mutations. And we can also make mutations. Well, I can't, but people at Oak Ridge can make mutations in the populus plants. Okay. So again, it's a matter of breaking something and then you try to fix it and then you can understand what it does. So this is a picture of a greenhouse, one of the greenhouses at Oak Ridge with the little populus growing, the seedlings. And also they have this really fancy thing at Oak Ridge. They have all the fancy stuff at the national labs where they can put a populus seedling into this chamber and everything about the seedling gets measured, like how much chlorophyll it has, what the area of the leaves is, like how much CO2 it's taking up and how much oxygen it's giving off, and all those kind of parameters. Because in the end, we want to know if we want to find bacteria that stimulate the growth of the plant and then understand how the bacteria stimulate the growth of the plant. And I always like try to really make this point when I'm teaching is bacteria are really, really, really small. Like 1 billion of them will fit in a teaspoon. And if a single bacterium was as big as we are, then we'd be 1,000 miles tall, which means like that if jets were flying around, they'd be like right at the level of our big toe. The bottom line of that is that bacteria are hard to see. And what scientists do is they're usually studying things that they can't see with their naked eye. And we have to infer whatever we're studying is doing by indirect means. And that's why we have so many different kinds of instruments and things in our labs. And so you'd think that we'd use a microscope a lot in labs so we could see our bacteria, but actually we hardly ever use a microscope. We just grow them up and do stuff to them and get them to grow as colonies. But usually we don't see them when we're studying them. We study things they do. And infer who they are from that. So, but there's one person at Oak Ridge named Jenny Morell, here she is, who does look at the populus bacteria. And she's studying one that's shown here. And this is a 2-micron scale. A micron is a millionth of a meter. So she can see them under the microscope. And then she can grow them in a colony and see different colonies. And she can put a colony on the right kind of growth medium and see that it solubilizes phosphate, which is a good thing because remember the plants like to take up solubilized phosphate. And also this bacterium that she works with a lot makes indole acetic acid, which is a plant hormone. So it stimulates the growth of plants. So she has like a great bacterium to start with. And it also here's a little populus seedling, okay? And if you look at the tissue of the populus under the microscope, which Jenny has all fancy microscopes to do, this is an uninoculated control, but after she inoculates the seedling in the soil with the bacterium that she's interested in, you can see, and her bacterium is tagged with a green fluorescent protein. You can see the bacteria inside the roots, okay? So she knows how many bacteria she has inside the roots. So this is really great. And so she can then start to study what the bacteria are doing in there. But the problem is that she infects the bacteria and then she sees them. But what she wants to do is, that's just a time when she has to sacrifice the plant, and it only provides a snapshot of what's going on. What she wants to do is study the whole dynamics of what the bacteria are doing in there. So she wants to develop, Jenny wanted to develop a method of looking at the plant microbe interaction without having to destroy the little plant. So then, this is another thing that they're really good at, the national labs, is fabricating things. So they fabricated these 3D little chambers, and they're the size of a microscope slide. And see, they can grow the little plant in the chamber, and you can see its root growing in this agar medium. And there's little things coming in to feed the plant. And she can maintain the plants for a little over 7 days. And so this is actually a wheat root. I think wheat have really good roots. So she started with wheat. And so this is her bacterium growing and colonizing the wheat roots. And here you can see, so this is a little rootlet here, but these are like individual plant cells, like from here to here. It's cut across and across. Let me see, will that work? No, okay. So you can see this is an individual plant cell, and you can see their bacteria in it. So she can study that and how they move around, and you can see them swimming around in there. It's kind of cool. So she can visualize the bacteria inside the populus, and by tagging them with something called a green fluorescent protein, which was discovered in a jellyfish off in the waters off Friday Harbor. The jellyfish was discovered, and then it was discovered that it had this green fluorescent protein, which is now used all over the place in biology. Because if you shine a fluorescent light on it, if you shine the proper wavelength on it, it gives off this fluorescence that you can see. So what she did is then she'd set up her green fluorescent protein in such a way, it's a gene actually, she'd set up the gene in such a way that when the plant gets when the right signal comes to the gene, it turns on the green fluorescent protein. So in this way, she can explore signals that the plant might make by seeing when the bacteria start looking fluorescent. I hope that's clear. So one thing she wanted to do, so this is like, we go from high tech to low tech and back and forth, okay? So this is like, I love this kind of experiment. So you got your little plantlet growing in an agar petri dish, and you can see its roots. And this is all sterile, pretty much sterile, okay? And then we put little strips of filter paper over them. And here is a filter paper with her bacterium in it. And they're kind of dull looking when you look at them under the microscope. But here are bacteria in the filter paper that's right across the plant root. And something in the plant root is turning on their green fluorescence protein. So they're becoming fluorescent. And so we call these reporters. We're reporting on the chemical environment of the root by looking for a response in the bacterium. And the response, we have certain elements hooked up to the GFP, and we can see the GFP as a response. So this is one way we can look at the chemical environment, but then we have to actually figure out what the chemicals are that are doing this, and that's a lot harder. Then another thing that she does is she puts her bacteria on these little filter paper discs, and then this is all full of bacteria. You can't tell. Okay, and then she takes her little discs and she puts them on like the root base and the mid root and the root tips of this little plantlet. Okay, and then she incubates for a week, and then take off these little filters, and the bacteria have been responding to the plant over this time. Okay, so we're going from this low tech, and then people in the project that have these really, really fancy instruments can take these tiny bacterial samples and figure out what the composition of the bacteria now is. So they look to see what their protein makeup is, okay? And so these guys will now have these bacteria. They're still the same, basically the same bacteria, but they'll have different proteins in them than these bacteria because they're responding to what the plant's telling them to do. So that's the kind of stuff we do. So that they do. So that's like really sophisticated instrumentation to look at the composition of the bacteria, going from this very unsophisticated setup. So that's all I really want to tell you, to give you a flavor of the kind of research that can be done to understand plant microbiomes. So we're just like little babies learning how to walk. So we're starting to understand plant microbiomes and interactions between plants and their microbial communities that they host. But we have a long way to go. And this project isn't the only project that works on this. You know, this is getting to be a very competitive area. Lots of people are starting to work on it. I haven't talked about fungi, which of course are what mushrooms are. But before a mushroom becomes a mushroom, it's just this net of stuff in the soil. And some fungi infect plants. And today I just showed you some examples of things that we can do with a single bacterial strain interacting with Populus. But we also, the PMI project also includes studies of synthetic bacterial communities. So we'll put, I keep saying we, but I'm not really doing any of this. So they put like 5 different bacteria together and infect the populace and see which bacteria win out. And they have a 10-member community of bacteria, and all of them infect the plant. But how do they interact with each other? And does the plant do better with all 5 there, or just one of them, or what? And there we even have a 125-member community, so 125 different kinds of bacteria that will infect the Populus seedling with all at once, and they'll get inside the Populus, and then what do they do? You know, we don't know. We got to figure all this stuff out. And and then we want to understand signaling between the Populus and the bacteria, which is usually by chemical signals. So that's really all I have to say. I hope I didn't go too fast, and I'd be happy to entertain any questions about anything, just about the DOE in general or research in general or anything about plants. But I just want to thank you very much for inviting me to share a glimpse of the types of stuff that we do. So thank you.
Thank you very much, Carrie. We do have some interesting questions.
Okay.
So the first one, I think you kind of answered as you went along, but I'll go ahead and ask you. Kim Clarkin wanted to know why Oak Ridge is interested in cottonwood and their bacteria.
Oh, yeah. Yeah. Well, I think that I talked about the biofuels thing, but also the DOE is really, well, there's a few branches of our government that do work on climate change. And we can't really say right now that that's what we're doing, because you know why. And so a lot of this kind of work really is, part of it is to really understand these plants and their dynamics and their interaction with sort of our atmosphere, so that we can be in a position to take advantage of plants to mitigate the effect of climate change, because plants absorb carbon dioxide, basically. So that's another reason that they study them.
Great, thank you. Actually, the second question was an interesting comment. There was a picture early on, kind of an along the road picture. And
Yeah.
Diane Doss is hypothesizing that it was the South Fork of the HOH, the Ho. Is that a?
Yeah, yeah, or it could be. I have to ask Sharon. Yeah.
So thank you, Diane. Then Yanka asked if there were reasons to use the populus for biofuels rather than going in the direction of algae.
Oh, yeah. Well, they do. Yeah. So I should have expanded on that more. There is a lot of research being done on algae too. But I think that the DOE is throwing, well, like BP was doing a lot of research on algae, for example. But I think, and the DOE used to do quite a lot of research, and they still do some, but I think they've decided that they should go to the plant biomass, because there's kind of a lot of problems with algae, growing them up in a big enough quantity to get the amount of biofuels out of them that you need. And you have to really take care of the algae. Whereas, populus, you don't really have to, you can plant them and then you don't have to do much to take care of them.
Great, thank you. I think you also answered this along the way, but Marcia wanted to know if all populous communities have similar microbiomes.
That's a great question. And we didn't know the answer to that when we started. And we think now that they do have kind of all the same microbiomes in general. But what we don't know is if they have some small percentage of microbes that are kind of specific to a particular species or a particular variety of populus. We don't know that yet, but in general, they all have kind of the same types of microbes, which are very different from the kinds that we have, that humans have. And I don't know, and there's going to be differences between different plants too, different species of plants, because different species of plants excrete very different kinds of compounds out of their roots.
Okay, thank you. Then Erica wanted to know if you're modifying the cultivated plants, I guess, to knock out their ability to reproduce so that they cannot mix with wild populations.
Oh, did we do? Oh, yeah. I don't think they've knocked out that ability. I don't really know how to answer that question, but
Okay.
I think it's a good one and one that I'm hoping they're paying attention to. Yeah.
Okay. Then Clinton Cho asked to me a baffling question. He wants to know if you carry out Koch's postulates procedures, KOCH.
Mm-hmm. We try to, yeah. So that means that you infect the plant with a bacterium, and the bacterium does a certain thing to the plant. And then you can isolate the bacterium out of it and then reinfect, and it has the same effect. I think that's Koch's postulates. I should know that. Yeah.
Okay.
So we
Well,
try to do that. Yeah.
I've learned something,
Mm-hmm.
something bonus. Then Kevin Webb, who appears to be writing from Thailand, says, thank you, this is fascinating. And then he goes on to say he's 69, but he did a part-time degree in his 20s in chemistry, so he's following okay.
Good.
And Barbara Bruel complimented you that it's very interesting.
Thank you, Barbara.
Brown complimented you that about the time you said you veered back and forth between high tech and low tech, that it's actually really helpful when you go back and forth from high tech to low tech because it makes it easier to follow for a non-specialist. And I agree with that.
Well, I think the low tech are the most fun. Yeah.
I like the little stickers and stuff.
Yeah.
Then there was a question that kind of opened a discussion. Cliff Cantor wanted to know what are synthetic bacteria. And,
oh, yeah.
People suggested that it's a synthetic community of non-synthetic bacteria.
That's right. They call them synthetic communities because we mix them together. But the bacteria we mix in are regular bacteria that we originally isolated from the populus. Yeah, that's a good question.
Thank you. Then Rebecca Adams wanted to know what you think would be some implications and uses of this research. Improved seed stock for reclamation or restoration, forestry, biomass production, or is it simply to understand the processes?
Well, our goal is to just understand the processes, but we're hoping if we understand them better, then we can use populus more efficiently to do land reclamation or to grow them in places where other plants can't grow very well. We can't, it's very controversial if you're going to genetically engineer the plant and genetically engineer their microbiome. And maybe that's something that would happen way in the future. But if we engineered their microbiome, we'd have to engineer some things so their microbiomes couldn't escape into the environment at large. But at least we can understand like factors that are helpful to the bacteria and helpful to the plant so that they can interact with each other optimally for each other's benefit.
Great, thank you. Here's an interesting question. I've come to understand that there are a lot of bacteria that can't be cultured on, is it pronounced agar?
Yeah. Uh-huh.
So do you have any way of assessing how much diversity you might be missing?
That's another great question. That's true, that there are things we can't cultivate, but what we can do is something called metagenomics, where we go into the plant and isolate bacterial DNA from the plant. This is really hard to do, very technically difficult, but they've solved the problem. of isolating bacterial DNA from the plant, and they can isolate bacterial DNA from the soil around the roots, and they can sequence that. And so from that, we can, there's certain genes you can sequence and it tells you what kind of bacterium they're from, roughly, you know. And so we can see, we can kind of get an idea of what we've missed from doing that. And we've done pretty well. We think we've isolated most of the major groups. Again, there's probably some bacteria that we haven't been able to grow. But we think we've got pretty good coverage of them. So that's the good thing about these plant-associated bacteria. They seem to be a little bit easier to cultivate than ones that grow in some other places.
Great, thank you. Here's a question from Shelley. Just as a little bit of background, we've had a couple of really interesting complementary lectures to yours on mycorrhizal fungi.
Oh, yeah.
In their behavior in plant roots and stuff.
Mm-hmm.
So Shelley's question is, are bacteria and mycorrhizal fungi basically performing the same services as far as the plant is concerned, or is there something that distinguishes them in the broad sense? Is there any evidence that plants might favor bacteria over fungi or vice versa?
I think that plants, no, I think that plants and fungi grow together very intimately. Fungi are called eukaryote. The fungi are more like we are. And bacteria are much more different. Like we and fungi are much more closely related than bacteria are to either of us. So the fungi and the bacteria have really learned to grow together well and intimately, and I think they help each other a lot. And we have a whole nother part of the project that studies that part of it. And they do all kinds of, we know a lot about how they signal to each other and how they help each other. And that's actually the part of the project that's probably the group has made the most progress on.
Great, well, thank you very much. I'm afraid I'm missing some questions here, but it's 8 o'clock.
Okay.
And I think this was a fascinating presentation that opened up a whole new world to me. So any parting words, Carrie, before I sign off?
Well, I just want to thank you for having me. It was really fun putting this talk together. And thank you for your great questions, excellent questions. So I guess we can sign off and either go to the news hour or not, depending on our dispositions.
Depending on our fortitude.
Yeah.
So thank you all for attending. I'm going to stop recording and end the meeting. And thanks again to Carrie.
Okay, thank you.